Today, we're diving into a case that has puzzled scientists for decades: pancreatic cancer. Think of it like a genomic detective story, where we're following clues hidden deep inside a tumor's DNA. And a really groundbreaking new study has given us a whole new set of forensic tools, letting us see this cancer in stunning detail. And what they found, well, it changes the entire investigation.
So first things first, let's just face the reality of the case. The 5-year survival rate for pancreatic cancer is a tough 13%. Now that's not just a number on a screen, it's a measure of urgency. It tells us exactly why we need new ways of thinking, new clues, and a much sharper view of our target. All right, here's our investigation plan for today. We're going to start with the problem we've been facing: cancer's blurry blueprint. Then we'll introduce the amazing new magnifying glass that's changing the game. After that, we'll uncover 3 major clues: a huge plot twist with the KRAS gene, 2 totally different origin stories for BRCA2 cancers, and finally, how the enemy is constantly evolving. Then we'll put it all together.
Okay, so for years, we've had this one major problem. Studying a tumor has been like trying to identify a single person of interest in a massive, out-of-focus photograph of a crowd. Sure, our methods gave us an average picture, but the key to cracking any case is always in the details, right?
And this analogy just nails the difference. Traditional methods, or bulk sequencing, are like throwing an entire tumor into a blender and analyzing the smoothie. You get the average genetic flavor, but you completely miss that there was 1 strawberry, 10 blueberries, and maybe a rogue piece of lemon in there. Single-cell sequencing, on the other hand, that lets us examine each piece of fruit one by one. We can finally see the individual rogue cells and understand what the tumor is really made of.
So let's meet the hero of our story, our new high-resolution forensic tool. It's called single-nucleus DNA sequencing. This is the incredible technology that lets us zoom in from that blurry crowd photo to a crystal-clear headshot of every single cell.
And the scale of this investigation is just mind-blowing. Researchers analyzed almost 140,000 individual cells from 24 different patients. And they weren't just looking at the primary tumor, no, they tracked the cancer's entire journey as it spread through the body. They basically created the most detailed evolutionary map of this disease we have ever seen.
Now for our first big break in the case. For a long time, a gene called KRAS was considered public enemy number 1. It's this master switch that tells cells when to grow and divide, and everyone thought it was the main driver of pancreatic cancer. But this new detailed view, it revealed a major plot twist.
And this brings us to a really crucial question. We've built therapies, entire fields of research, really, all around the idea that KRAS is the essential engine of this cancer. But is that always true?
Well, the answer is kind of shocking. In nearly 43% of the cases, the researchers found large populations of cancer cells that have actually lost the founding KRAS mutation as the tumor evolved. Just think about that for a second. The cancer started with this mutation, but later on, some of its descendants just ditched it.
And here's why that is so important. If a cancer can evolve to live without its original driver, then a drug that's designed to target that specific driver might stop working, or it might never have worked in the first place for those cells. This is a massive clue for understanding and hopefully overcoming drug resistance. The cancer is way more adaptable than we ever thought.
All right, let's move on to our second clue. This one involves the famous BRCA2 gene, which normally acts like a DNA repair mechanic in our cells. Now, we've known it's linked to cancer risk, but this study found something new, that the timing of when it fails creates 2 completely different types of cancer.
So in the first origin story, the early hit, losing both copies of BRCA2 is one of the very first things that happens. It's the opening act, and it kicks off this really chaotic process. The cancer doesn't follow the usual script at all. Instead, it's driven by these massive genomic rearrangements, all because of that early instability. But then you have origin story number 2, the late hit. Here, the cancer starts out on the conventional path, driven by the usual suspects like KRAS. It's only later, once the fire is already raging, that the second BRCA2 gene fails. So in this case, losing BRCA2 isn't the cause. It's more like an additive trait, an upgrade the cancer gets along the way. And this distinction is vital, because a cancer born from chaos might need completely different drugs than one that just added a new weapon later on.
And that brings us to our final clue. And honestly, this might be the most revealing one of all. It's not about just one gene, but about how the cancer relentlessly disables one of our body's key defense pathways over and over and over again.
To really get this, we need a quick concept from biology: convergent evolution. It's the idea that different groups can independently find the same solution to the same problem. Think about how bats, birds, and insects all separately evolved wings so they could fly. Well, it turns out cancer cells do the exact same thing.
What the study found was incredible. Within the same tumor, different groups of cells were all figuring out their own unique ways to sabotage the same defensive system, a pathway called TGFβ, which normally puts the brakes on cell growth. So some cells would get one mutation, while a totally separate group would get a different mutation. But the outcome was always the same: that defense was shut down. This shows us there is this immense, constant pressure on the cancer to overcome this specific barrier as it grows and spreads.
So we've gathered our clues. We have a main suspect that isn't always essential, we have two different origin stories, and a relentless, evolving enemy. Let's put these pieces together and see what this new, high-resolution picture of pancreatic cancer is really telling us.
Here are the three crucial takeaways. First, a cancer's dependence on the KRAS gene is absolutely not a given. Second, for genes like BRCA2, when something happens, fundamentally changes the nature of the disease. And third, cancer is constantly evolving under pressure, finding multiple, different paths to the same deadly goal.
You know, a quote from the researchers themselves just sums it all up perfectly. To solve this case, you have to look at the individual suspects. Bulk sequencing, that blurry photo of the crowd, it could never have revealed these clues. It couldn't tell us the order of events or see the different factions evolving inside the tumor.
And this whole investigation leaves us with one final and really powerful thought. By using these new forensic tools to reconstruct a cancer's detailed history, to basically read its evolutionary diary, cell by cell, we are getting closer than ever to a profound new ability: the power to not just understand its past, but to finally predict and maybe, just maybe, control its future.